Welcome to this explainer. I am so thrilled you're turning in, because today we're embarking on a genuinely fascinating journey. We're going from the macro level of biological taxonomy all the way down to the micro level of, well, literally digging in the dirt. We're going to unpack the biological architecture and the classification of Annelida, and specifically, we're zooming in on one of the most underappreciated creatures on the entire planet, earthworms.
So think about this for a second. What is a tube within a tube? Breathes entirely through its skin and acts as the master engineer of the Earth's soil. I know it sounds completely alien, right? But it's literally right beneath our feet, working tirelessly 24/7 to keep our ecosystems functioning. Okay, let's dive into this. Here's our roadmap for today. We'll kick things off with a welcome to Phylum Annelida. Then we'll check out the earthworm's family tree. After that, we'll explore earthworm diversity and lifestyles, look at collecting earthworms in the wild, and finally wrap up with preserving specimens for study.
Welcome to Phylum Annelida.
First up, let's look at the name itself, Annelida. It actually comes from the word annule, which translates to little ring. You know, if you've ever looked closely at an earthworm after a heavy rain, you've definitely seen these little rings. And that brings us to a really crucial biological term, metamerism. Basically, metamerism means true external and internal segmentation. It's not just some surface pattern. This segmented structure runs right through their internal organs, and it's present in both the developing embryo and the fully grown adult. This little ring architecture, it's absolutely the defining characteristic of these incredible segmented worms.
Now, let's quickly demystify some of the biological jargon here. Earthworms have triploblastic germ layers, which, honestly, simply means their bodies develop from 3 distinct embryonic cell layers. They operate on an organ system level of organization. So yeah, they have actual working physiological systems just like we do. They feature bilateral symmetry, meaning if you drew a line right down the middle, the left side perfectly mirrors the right side. But here's the coolest part. They possess a true column, specifically a schizocolumnate. This is a mesoderm-lined cavity between their body wall and their gut wall. Think of it like a smaller straw floating inside a slightly larger straw. That's the famous tube-within-a-tube body plan that gives them their totally distinct physical structure.
And you know, this body plan brilliantly illustrates the incredible adaptability of annelids across some wildly different habitats. The phylum is actually broken down into 3 main classes. First up, Polychaeta, which are mostly marine worms like the Nephridia. Fun fact here, many of these marine worms use lateral appendages called parapodia to paddle and move around in the water. Pretty neat, right? Then we have Oligochaeta, which houses our familiar terrestrial earthworms. And finally, Hirudinea, the freshwater dwellers like Hirudinaria, which, let's be real, you probably know better as leeches.
Section 2, the earthworm's family tree.
You can kind of think of this systematic position as finding the earthworm's exact address in the massive, sprawling city of the animal kingdom. So they live in kingdom Animalia, Phylum Annelida. Their specific neighborhood is the class Oligochaeta, which literally means few bristles. Then we go down their street to order Haplotaxida, marked by that elongative shape and classic pinkish-brown color. Their specific apartment building is the family Megascalachidae, or Lumberschidae, known for having numerous setae, which are these tiny, hair-like bristles. And finally, we arrive at the exact apartment door, genus and species, like Pheretima posthuma or Icenia fida. Notice how every single step of this taxonomy is directly tied to a physical trait.
But how do they actually function biologically out in the world? Well, for starters, earthworms are hermaphrodites. In biology, we call this being monoecious, meaning both male and female sex organs are present and fully functioning within the exact same body. And honestly, equally fascinating is how they manage to breathe underground, because they don't have lungs. Instead, they rely entirely on cutaneous respiration. This means they literally breathe right through their moist skin, which allows vital oxygen to diffuse directly from the dirt around them right into their bloodstream.
Section 3, earthworm diversity and lifestyles.
Okay, are you ready for this number? 6,000. There are over 6,000 vast and incredibly diverse species of earthworms worldwide. If you thought an earthworm was just a simple pink wriggler you find on the sidewalk after a storm, well, think again. They vary wildly in size, behavior, and habitat. We're talking everything from deep soil dwellers all the way up to species that have adapted to live high up in the canopies of trees. Absolutely wild.
Now, what's really interesting is how ecologists actually categorize all this massive diversity. We group them by their lifestyle, which dictates exactly where they live in the soil column.
First, we've got the epigeic worms. These are your surface dwellers, mostly hanging out in leaf litter and decaying logs. Next are the endogeic worms. They live their whole lives just below the surface in the rich upper soil layers. And finally, the anisic worms. These are the deep burrowers that create extensive permanent vertical tunnel systems way deep into the earth.
So why does all this matter? Well, the big takeaway here is that earthworms are irreplaceable ecological engineers. They play a truly crucial role in soil health, fertility, structure, and unique nutrient recycling. By burrowing and feeding, they physically aerate the soil and chemically break down organic matter. Whether we're talking about native species maintaining a local forest or exotic species specifically used by humans in commercial vermicomposting, they are the absolute baseline of a healthy terrestrial ecosystem.
Section 4. Collecting earthworms in the wild.
Okay, so if you're a biologist out in the field, how do you actually go about finding and extracting these populations to analyze them? We rely on a few primary sampling methods. There's hand sorting and chemical extraction, which is also known as using a vermifuge. Then we have soil pit sampling, which involves digging highly specific 20 by 20 by 20 centimeter measurement pits, usually during the wet seasons. And finally, habitat inspection, where researchers actively dig through specific microenvironments like leaf litter, debris, or compost heaps.
Let's actually compare the two most contrasting methods here. Hand sorting is essentially getting your hands dirty, digging and manually sifting through the soil. It is incredibly labor-intensive, but it's highly effective for those small, shallow-dwelling epigeic worms we just talked about. But what if you need to study the deep-burrowing anisic worms? I mean, you can't exactly dig a 6-foot trench for every single sample. That is where chemical extraction comes in. Researchers pour diluted mustard solutions, or sometimes formaldehyde, into the soil to act as an irritant. It rapidly drives those deep-burrowing worms right up to the surface for easy collection. So, the technique you choose? It depends entirely on the specific lifestyle of the worm you're trying to study.
Section 5. Preserving specimens for study.
Once you've collected them, the very first step is always to gently wash the worms. From there, your protocol splits based entirely on your study timeline. For short-term handling, you simply want to keep the live worms comfortable in cool, damp, well-aerated soil so they behave naturally. However, if your goal is to add them to a museum catalog, or maybe build a permanent reference collection for long-term anatomical study, you need long-term preservation, and that requires chemical fixing.
Let's move to and see how this builds into a rigorous lab protocol for long-term preservation. Step 1 is absolutely crucial. You must anesthetize the specimen in 70 to 100% isopropyl alcohol for just a few seconds. Why? Well, because if you skip this, the worms will instinctively contract and knot up, making their anatomy basically impossible to study. Step 2 is to fix the now relaxed specimen in formalin for 24 hours. That preserves the cellular tissues. And finally, step 3, you transfer them to a permanent storage solution in alcohol.
As we bring this explainer to a close, let's map out the specific earthworm species that researchers and hobbyists tend to collect most frequently. You'll probably recognize some of these common names. There's Lumbricus terrestris, which is the standard common earthworm you see everywhere. Then there's Icenia fetida, famously known as the compost worm, super highly prized in agriculture. And Eudrilus eugenii, the much larger, highly active African nightcrawler. These guys are the undisputed stars of the soil biology world.
Which leaves us with this final, profound question to consider. Without these hidden, skin-breathing engineers constantly recycling our world beneath our feet, what would happen to the soil we rely on to survive? The next time you walk across a park lawn or a forest floor, just remember the millions of tiny little rings working tirelessly in the dark beneath you. Thanks so much for joining me on this explainer, and keep staying curious about the incredible hidden mechanics of the natural world.